So this is continuing photosynthesis. This was the light-dependent reaction. Ultimately, the process is building fuel, but it's building a stable fuel. Is that these are really usable, but they're not long-lasting. You got to use them pretty much just after you've made them. But that's what's going to happen. The second part of this uses this input. And that, of course, is the light-independent reaction. This is where the carbon dioxide goes in the NADPH, and the ATP. Now, I hope this is big enough on whatever screen you're using that you can see at least enough to be able to make it out when I point at it. So this is the Calvin cycle. It uses carbon dioxide. Now, anytime you have a cycle, your entry point attaches to something in the cycle, changes it, but eventually you get it back. This is one of the, remember I said I spent nights putting this onto huge sheets, drawing all the molecules. This was the killer. Yeah, let me get that show up better. You have 3 molecules of a 5-carbon compound here. You pick up 1 carbon dioxide. Now you have 6 molecules of a 3-carbon molecule, 6 3-carbon molecules. ATP comes in. ATP comes in, NADPH comes in. You're basically energizing the molecules. You've got phosphoglycerate. Then you've got biphosphoglycerate. You're getting the phosphates off of here. You're energizing this with the hydrogens. You lose 1, you wind up with 5. It changes the molecule again, changes the molecule again. You're back where you started from. This is the 5-carbon molecule you started with. This is where the carbon dioxides go in. I know. You don't need to know it in this level of detail. This sticks a carbon in there that didn't used to be in there. That's why they call it carbon fixation. This sticks the molecules together so that you get a particular type of 3-carbon molecule that can then be stuck together to make glucose. Remember I said that happens slightly outside of the loop? And then you basically regenerate an energized version of a molecule that can pick that up again. When we look at aerobic respiration, we're going to start with glucose. We're going to split it in half. We're going to feed it into a cycle. We're going to run it through an electron transport chain, which I just realized I should have mentioned as a big part of the light-dependent reaction. Sorry. Don't you hate it when people do this, stick something between the lines when you've already written stuff down? Terrible thing is, it's in my notes and I missed it. Because I was thinking, oh wow, that should be in my notes to make sure I don't skip over it. Well, it was in my notes and I missed it. That happens sometimes when I'm looking up and looking down, looking up and looking down, and wandering around in front of the camera. So ultimately, you've got the Calvin cycle. makes 3 carbon compounds. You don't need to know the names of those. And that's used to produce glucose. Ta-da! Final step. It's what you want to get to. And that's kind of, sort of, photosynthesis. There are variations. I'm just going to, there's 2 of them mentioned in the book. I'm going to just talk about one of them here. C4 uses 4 carbon compounds with 4 carbons in them, not 4 carbon compounds, but carbons with 4 carbons inside the compound. It turns out that it can be done more efficiently with less water. So you tend to find this in hot or dry conditions. In those rare situations where carbon dioxide is not readily available, C4 photosynthesis is more efficient, so you'll find it under those conditions as well. I decided to back off of putting that down. There's a chemistry at work here that reflects what's going on. When you're a plant growing in hot, dry conditions, you need water, right? That's going to supply the hydrogens for the light-dependent reaction. But it's hot. You need carbon dioxide in order to put the carbons together. Typically what they do is they flush the leaves with carbon dioxide from the air and water from the ground at night. They open their pores to let the carbon dioxide in. And when the pores are open, water's going to leave. The air's dry, transpiration is going to happen. You're losing water that you otherwise would have used. So, but it's cooler, and so you don't lose as much. Daytime comes, you slam shut all the pores, and then you go to work. So C4 plants sometimes don't do photosynthesis through the entire day because they, if they open up their pores to let in more carbon dioxide, they lose the little bit of water that they've got. You do wind up with an abundance of oxygen in the plants, and there's chemistry at work to keep radicals from being damaging, because you can't open the pores to let the oxygen out either. You do use a little bit of oxygen in respiration, but plants are not that energetic, so a lot of the oxygen is just going to be, released, except you got to be able to release it. This brings us to respiration. Or wood. If I can find that part of my notes. There we go. Fortunately for Sparky, Zeke knew the famous Rex maneuver. Don't you hate it when you get a cat stuck in your throat? You can't breathe. Now remember, this is cellular respiration. This is pulling molecules apart and using the energy to do stuff, which almost always involves pulling molecules apart, moving that energy into something that's easier to use, and using that stuff, ATP usually, to do the things that the cell needs to do. It doesn't always use glucose, but the classic type of respiration uses glucose, so we're going to basically do glucose as the main input. There's a bunch of different types of respiration. The one thing that they have in common, other than that they pull molecules apart and get the energy out of them, is that at the very end, electrons have been used to make ATP, but they have lost so much energy that they're not usable anymore, but then you got to get rid of them. Remember, electrons are ferried around by hydrogen. You need a terminal, an at-the-end electron acceptor, in order to run the process. Now, aerobic respiration uses oxygen as the electron acceptor. That's why water comes out at the end. But that's not the only thing that you can use. Sulfur. If you've ever been in a swamp and you stepped into the muck at the bottom and it was this black, oily, sticky stuff, and when you pulled your foot out, you could smell rotten eggs, is that there's anaerobic sulfur-fixing bacteria in there. They're using sulfur as their final electron acceptor. Nitrites, the undergravel filter bacteria will produce nitrates, and then there's some that will take the nitrates and convert them to nitrates as well. There are some that use carbonates. There's an excellent chance that the very first respiration systems may have used iron. It was available because aerobic respiration hadn't evolved yet. Once aerobic respiration was widely disseminated, the iron reacted with the released oxygen, became iron oxide, and settled out of the water. So you don't find that many places where iron is still used. And as I said, oxygen. If you're using anything other than oxygen, you're doing anaerobic respiration. And this is, you have to think historically, when people started to figure out how this worked, what exactly are we breathing? What exactly are we using? Oh, we're using oxygen. Great, that's respiration. Then they just started to, they made their first discovery of things that seem to be able to live in a totally non-oxygen environment. Well, they're not using oxygen for respiration, they're using something else. They're not, oxygen is aerobic, these are anaerobic. It took a long time to figure out there's a whole bunch of different types of respiration. And there's the oxygen and the sulfur and the carbonate and the iron, but they lump them together. It's kind of like vertebrates and invertebrates. Vertebrates aren't even a whole phylum, but they're our group with their nice backbones. So there's our group, and then everybody else, the 20 other phyla over here that are all lumped together as invertebrates. I'm going to talk about a little bit about anaerobic organisms that have impact in human situations. These are organisms that can survive outside of the presence of oxygen. There's not a huge number of environments where oxygen isn't prominently featured. Now, some anaerobic organisms are actually poisoned by oxygen, is that they can't exist except in some sort of dormant form when they're exposed to oxygen. If you want to kill a termite with a very strange approach, you can take a bunch of termites and put them in a chamber and pump a bunch of oxygen in at high pressures. what digests the wood carbohydrates for termites are anaerobic organisms that live in their guts. Their guts are no oxygen environments, and these guys pass from termite to termite in a dormant cyst form. They can survive exposure to the air there. But if they're out and active in the gut, you force oxygen into the gut by putting them into high pressure situations. The oxygen isn't at a toxic level for the termites, but it winds up getting into their intestines at a toxic enough level that it kills their little buddies. And then they keep chewing and chewing and chewing, but it doesn't matter, is that they starve to death because their actual digester helpers have been killed by the oxygen. I know that's a weird story. But you do find anaerobic organisms as kind of these fringe organisms in these strange places. We have them in us. Our guts are anaerobic ecosystems also. And we have some, most of our little buddies are not toxic. Oxygen is not a toxic thing to them, but they're perfectly happy where there's no oxygen. And of course, as we're going to see, aerobic respiration is much more efficient. It's a better system in a lot of ways to anaerobic. So aerobic organisms tend to outcompete. They have more energy, anaerobic organisms. So anaerobic organisms kind of exist where there's not the aerobic competition, whether or not oxygen is a problem for them or not. So I'm going to give you a list of human impact from anaerobes. Diseases from anaerobes, oops, you can't see any of that. Diseases from anaerobes tend to fall into two categories. There are things that we take in that are anaerobic organisms that can live in our colons, in our low oxygen environment. But they don't necessarily get along with the buddies that we have already in there, which are going to wind up on the list eventually. Something like salmonella. Salmonella causes a lot of digestive issues, primarily because it throws your own system way out of whack. But then there are a lot of anaerobes that themselves can't really live in us or live in very limited circumstances. But what they throw at us are chemical byproducts, waste materials, or other kinds of toxins. Tetanus. Tetanus is a soil bacterium. It tends to be active deep in the soil in low oxygen environments. But it spreads through spores. The spores settle on things. You get a puncture wound, typically in a fairly thick part of your skin, because your skin has oxygen coming in from your blood and oxygen exposure from the air. But if your skin is thick enough, there's a mid-range where there's not a lot of oxygen. Tetanus bacteria are only going to become active in a fairly low oxygen environment. So you poke this thing in your skin, you get some of these spores into that low oxygen mid-range, and they start to reproduce in there. Is that, they're decomposers, so they're taking in nutrients from the materials around them, your skin. In the process, they release a pretty potent toxin that diffuses out into your circulation and messes with your muscle chemistry, locks it up. That's why tetanus is called lockjaw. One of the early symptoms is you start to feel the tightening and you have to feel it in your jaws, but eventually it affects your breathing. It's pretty bad. The vaccine for tetanus is actually the toxin twisted enough that it's not actually the toxin, but your immune system forms an antibody to it that will attach to the actual toxin before it can do its nasty work. And sometimes that's what antibodies do. They cap what would be an active molecule and deactivate it. So there's botulism. You get a small crack in a can. when food's canned, it's boiled, all the oxygen is driven out of the liquid, they put it in a sealed can, and it's sealed. There are botulism spores in the environment. A small opening in a cracked can doesn't let in a significant amount of oxygen, but sometimes spores of botulinum bacteria can get in there. And now they're in a low oxygen environment. They're happy as little, anaerobes. They start to chow down. They release a toxin into the material in the can. So you take this punctured can or this cracked can, dented really, and that dent can open a very small, an opening so small that the liquid doesn't leak out, but things can get in. You take your soup or whatever, your canned veggies, you put them in a pot. Now, you can deactivate this particular toxin by heating it for a decent amount of time, but most people don't boil canned stuff for like 20 minutes. So if you haven't done that, the toxin is still in its active form and you get botulism from it. Pretty nasty. I'm thinking, looking at this list, thinking if I go on with the list, this is going to get to be a really long video. So I'm going to break this up and put the rest of it on the next video.